welcome to our guest lecture held by the Faculty of Life Sciences and Technology, Swiss German University. So we have here students from the Faculty of Life Sciences and Technology, and we have also alumni and lecturers, and also guests outside SU. Welcome to our guest lecture. So today we have two special professors from Japan who will share their knowledge to us in the guest lecture. So the first speaker is Professor Ichikawa. The full name is Professor Sosaku Ichikawa. So he is a professor in Tsukuba University, Japan. Welcome, Professor Ichikawa.
Thank you. Thank you for
Yeah.
your kind introduction.
So Professor Ichikawa is a professor in the Faculty of Life and Environmental Science in Tsukuba University. So they have also master and doctoral program in Tsukuba University. So he will also introduce the program to us before he start the lecture. And thank you for your time, Professor Ichikawa. And then the second speaker, we have also Professor Toshiyuki Kanamori. Welcome, Professor Kanamori. I have been knowing Professor Kanamori for a long time. When I was a doctoral student in Japan, long time ago, and then I was hired by him to work together to do research together with him in National Institute of Advanced Industrial Science and Technology in Tsukuba. So he is one of the top researchers in Japan, working in National Institute AIST is one of also the largest research institute in Japan, and I'm very glad to have him here in the guest lecture. And then he's also a professor in Tsukuba University, so you can introduce later. And then the topic today is related with drug delivery, actually, but actually Professor Ichikawa and Professor Kanamori can explain more about the topic today. So I would like to welcome Professor Ichikawa to start the lecture. So time is yours, Professor Ichikawa, please.
Yes, thank you very much. So, okay, I will share my screen. Yes. Okay. So, good afternoon, ladies and gentlemen, and thank you for participating these lectures. So, first of all, I would like to thank you, Professor Samuel-san, to give us the opportunity to give a lecture in your university. Okay, like this. So, okay, so firstly, I would like to introduce our master's and doctoral program. The name is the Tsukuba Life Science Innovation Program in University of Tsukuba. So this is Tsukuba in Japan. As you know, here is Japan. So this is the enlargement of the map of Japan. So Tsukuba is roughly 60 kilometers from the Tokyo. It takes less than 1 hour by train from Tokyo. It's very close to Tokyo. So in Tsukuba, there are 59 educational and research institutes. Also, it's well-known science city in Japan. So in this Tsukuba, So we have a campus of University Tsukuba here. You can, our campus is in this photo. This is very large. The 4 kilometer by 2 kilometer. It's very large National University of Japan. The staff of research and educational, the number of staff of research educational number is around 1,900, and the number of undergraduate students roughly 10,000, and number of graduate students is 7,000. So because of the National University of Japan, so the tuition fee is relatively cheap. As I written here, in Japanese yen, the annual tuition fee is 53 like this. The Indian rupee, in the Indonesian rupee here. So in here we have Life Tsukuba Life Science Innovation Program. We are calling TLSI. So we have a web page here. So if you're interested, please visit this web page. So in shortly, I will introduce our program. The master and doctoral program in the Life Innovation Academic Program of University of Tsukuba offer talented students from around the world the opportunity to enhance their unique experience in becoming catalysts for innovation in the society of introducing new way of thinking. So TLSI, our program, will provide great learning opportunities here and also abroad, encouraging students' interaction with leading world experts in the six fields written here of specialization. One is the disease mechanism field, another one is biomaterial field, another is bioinformatics field, another is environmental science field, another is the food innovation field, another is the drug delivery field. Students can learn the subject in these fields in our program. So our program is very unique curriculum because we have a professor not only from the university but also the National Institute and also a private research institute. So you can see the many logo of the institute in Tsukuba area. The professor belonging to these institute, they are teaching the subject in our program. So you can see here, National Institute for Advanced Industrial Science Technology. This, as Professor Samuel introduced, Professor Kanamori belonging to this AIST, and also he is the professor of our program in University of Tsukuba. So not our program, a professor in our program is not only from the Japanese institute, but also from foreign countries, foreign research institute. This is the map of the professors from abroad. The professor in, for example, Wageningen University in Netherlands and Oxford, ETH Zurich in Switzerland, University Montpellier, France, Center of Biotechnology in Tunisia, and Malaysia, and Stanford University and California University in US. So some professor will give the lecture from these university or research institute in our program. All education in our program in English, so we can accept all of the students from worldwide. This is some detail about the subject in 6 areas. We have 6 specialized subjects for in 6 special area here: disease mechanism and drug discovery and food innovation and environmental management and biomolecular engineering and bioinformatics. So student must take these basic subjects and specialized subjects, and also their own specialized subject they must take from here. For example, the student in the disease mechanism, they must take these subjects. In the student in the drug delivery area, they have to take these subjects. Then finally, they can get the degree in the, for example, these degree. For example, when student finish the disease mechanism area, they can get master of disease mechanism. And if student finish the drug discovery course, they can get the master of medical science. If they finish the food innovation, they can get master of food innovation. In the field of environmental management, they can get master of environmental management. If they finished molecular engineering, they can get master of bioengineering. If they finish the bioinformatics course, they can get master of bioinformatics like this. This is also the subject in the doctoral program. The student in doctoral program, all of students of doctoral program must take these basic subject and specialized subject. And also they can take a special subject in each field. So if you interested in, please visit our web page written here. Maybe you can find our web page by Google. Okay, that's all about the introduction our master and doctor program in University Tsukuba. So let's study in Tsukuba together. So if you're interested in our program, please send us email. I will reply your question. That's the introduction of our master and booster program. Next, I move to the contents of my lecture. Okay, today I will introduce.
I know.
Yes. Okay. Okay, I will move
it's okay,
to
you
our.
can continue.
So the title is Lipid-Based Micronano Dispersion System. So today, for firstly, I introduce by myself. So my name is Sosaku ICHIKAWA as professor of University Tsukuba, and also now, currently, I am a program leader of Life Science Innovation Program. So I get a PhD in engineering in the Department of Chemistry and Biotechnology at the University of Tokyo. After getting a PhD, I was a postdoc from 1995 at the National Food Research Institute at the Ministry of Agriculture, Forestry and Fisheries, Japan. This National Food Institute also in the Tsukuba University. So it means that from 1995 to now, I'm staying in Tsukuba. So after this postdoctoral fellow, I get the position of research associate in University of Tsukuba at 1997. Then currently I'm a professor in this same University of Tsukuba. During my belonging to the University of Tsukuba, I was academic guest in the Department of Material Science at ETH Zurich, Switzerland. During this 1 year, I stayed in Zurich to study about molecular assembly for drug delivery or to make some artificial cell and so on. So my research area is biochemical engineering, especially for lipid-based liposome and emulsion and nano-micro dispersion system for pharmaceutics, cosmetics, and food. So this is my background. So about the lipid-based micro-nano dispersion systems, the utilization of these lipid-based micro and nano-dispersion systems, especially in food and pharmaceutical and cosmetics industry, has gained wide attention due to its ability to enhance the stability of ingredient throughout the process of manufacture, distribution, and storage of food product, and also to control the release and bioavailability of ingredient through the digestion and absorption. So the formulation and characterization and property control of lipid-based micro-nano dispersion systems are an important issue to use for this field. So this is an example of micro-nano dispersion system. You can see this illustration. This is a typical oil droplet in water phase. It's called OW emulsion. So on the other hand, if the water droplet dispersed in oil phase like this, it's called W emulsion. The more complex system is like this. For example, this is the WOW emulsion. In this case, there is an oil droplet in the water, continuous water phase, and the small water droplet exists in this oil droplet. So we are calling water in oil in water emulsion. OWO is the inverse system. And also some modification of droplet can be done. For example, coated by polymer like this, and sometimes coated by solid particle. It's called pickling emulsion. And also sometimes incorporate some ingredient in the droplet or particle. The solid can be entrapped in the oil droplet. And also sometimes some droplet can be captured by the gel matrix. And also sometimes the droplet can be solidified to make stability. And sometimes the surface of droplet coated by shell to make a stability. And also there is another system, the lipid dispersion system called liposome. Later on, I will explain more detail about this liposome system. And also sometimes micelle and microemulsion can be used as a dispersion system. So many types of dispersion systems are used for examining to enhance the stability and control the release and bioavailability of ingredient. So today's my content, we have 3 topics These are my research topics in my laboratory, but today just I introduced the abstract of these research work. First topic is the formulation of lipid vesicle with high entrapment efficiency using a WW emulsion as a template. So the lipid vesicle is also called the liposomes, are compartment that are formed in water phase by just molecular assembly, no covalent bond of barrier forming lipid. Especially the phospholipid can form this lipid vesicle. This lipid vesicle can potentially used as a carrier of functional materials, model membrane, and artificial cell, because this lipid bilayer is the same as the bilayer biomembrane of the cell. Currently, also this lipid vesicle can be used for the microreactor and so on. These are potential applications of lipid vesicle as a carrier. Materials that can be encapsulated inside vesicle, especially hydrophobic material, can be encapsulated inside the lipid vesicle, and also the Also, the hydrophobic material can be captured in the lipid bilayer because this area is very hydrophobic. So the hydrophobic material can be captured in this bilayer. So this lipid vesicle can encapsulate hydrophilic and hydrophobic, both properties materials. This is some example the materials that can be encapsulated inside the vesicle. You can see the antioxidant and some enzyme and vitamin and so on. So there are many research, the encapsulation of some active compounds inside lipid vesicle. So this is the illustration how to prepare the lipid vesicle. This is a general preparation procedure of the lipid vesicle. Firstly, the bilayer forming lipid is solubilized in the organic phase like this. For example, many cases the people are using methanol or chloroform. After that, evaporate the solvent. We get the dry lipid film at the bottom of flask. So then this dry lipid film can be hydrated by addition water. Then make a vigorous shaking. This dry lipid film spontaneously make this lipid vesicle. Many layers lipid vesicle. It's called multilamellar lipid vesicle. Then when the hydrophilic materials disperse in water, these materials can be captured in this lipid vesicle, but just by chance. It means the encapsulation yield of this hydrophilic material is very low in general preparation method. So the issue to be improved in the vesicle preparation. So as I described you, the low entrapment efficiency for hydrophobic materials into the internal water phase of vesicle, and also the difficulty in the size controls with keeping high entrapment efficiency. So then Our object of research was the development of the preparation method of lipid vesicle with a high entrapment efficiency and a control size using emulsion as template. This is the step to prepare the lipid vesicle from WW emulsion. Firstly, we mixed water phase containing hydrophilic materials with the oil phase containing lipid to make a W emulsion, water droplet in oil emulsion. The hydrophilic material exists in this water droplet. The second, we make a WW emulsion like this. This W emulsion dispersed in aqueous phase to get oil droplet containing this small water droplet. This is a WW emulsion. After that, evaporate this organic solvent from this organic phase. So these lipid can not be removed by evaporation. So these lipid in oil phase surround these small water droplets. Then we can get lipid vesicle like this. The hydrophilic materials in the water droplets remaining inside this lipid vesicle through the process. That's why we get the high entrapment yield. This is a typical example of the preparation of our method. We make a W emulsion in step 1 like this. This is a water droplet containing green fluorescence materials. Then we make a WW emulsion using microfluidics like this. You can see the oil droplet formation through this microchannel. Then we get this oil droplet containing small water droplet. In the small droplet, water droplet, there is the green fluorescence molecule, so we can see the green droplet by microscopy like this. From this WW emulsion, we evaporate organic solvent like this. This is the continuous phase. This is the oil droplet accumulate the surface of the aqueous phase. After the organic solvent evaporation, we get this lipid vesicle suspension like this. So this is a microscopic observation during the formation from WW emulsion here. Then after the hexane evaporation, we can observe this kind of aggregate of lipid vesicle. After that, this small lipid vesicle disperse like this. So then we get the lipid vesicle containing some materials inside it. This is the size distribution of the initial water droplet. In this case, we prepared around 200 nanometer of water droplet at the first step. This is the size of water droplet in the first step. Using this W emulsion, we prepared lipid vesicle following step 2 and step 3. This is emulsion, W emulsion. This is a lipid vesicle. We can observe the lipid vesicle formation by freeze fracture stem like this. you can see the size of this lipid vesicle. Around diameter is around 200 nanometer, so it's comparable to the size of the initial water droplet shown in previous slide. The entrapment yield, for example, calcine as a fluorescence marker is 93%. For the 5-fluorouracil as a cancer drug is around 40%. This entrapment is relatively high compared to the other preparation method. This is the size distribution. It's the same. The size is the size of lipid-based prepared lipid vesicle as same as the initial size of water droplet like this. So we can control the size and we can achieve the high entrapment efficiency by using this method. This is a short summary of our first topic. So lipid vesicle with high encapsulation yield for the water-soluble compounds could be prepared by using WW emulsion as a template. And also the vesicle size was controlled by the primary emulsification process. So I think it can be used for the encapsulation of some drug or herbal essence or food ingredient in the lipid emulsion using this method. This is the first topic in my talk. The next topic is the layer by layer mulch coating of large oil droplet by polyacrylate dietary fibers. This is the layer by layer mulch coating of emulsion. So this mulch coating is one of the methods for modification of oil droplet surface properties. Mulch-coated emulsion were formulated by layer. It's called LBL, deposition, using electrostatic polymers onto the surface of the oppositely charged droplets through electrostatic attraction. Electrostatic polymers coating are formed by the repeating of the coating process a number of times using oppositely charged polymers. This is a schematic illustration of layer by layer coating of oil droplet. Firstly, we prepare oil droplet using some detergent, surfactant. Generally, the surface of droplet of emulsion is negatively charged. When we add the positively charged edible polymer in the system, that positively charged polymer adsorbed on the surface of oil droplet like this. Then we can get one layer of positive polymer on the surface of oil droplet. After that, we add negatively charged edible polymer in the system. Then the negatively charged polymer adsorbed on the surface of oil droplet like this. So we get the two layers of the polymer on the surface of droplet. After this, we can add the positively charged polymer. We can get three layers like this. This is a schematic illustration of layer by layer coating of oil droplet. So we focus on the very large oil droplet in the micrometer size oil droplets because the very large oil droplet is unstable compared to the submicron size of oil droplet. But regarding the entrapment capacity, large oil droplets are preferred due to the increased volume. So we aim to determine if it's possible to increase the stability of large oil droplet larger than 10 micrometer size by this layer by layer deposition using cationic chitosan as a polymer and anionic carboxymethyl cellulose as polyelectrolytic dietary fibers. Firstly, we prepare the oil droplet using microchannel technique. This is an illustration of microchannel. We push out the soybean oil as an oil droplet, as an oil phase through this channel. This channel made by silicon etching. So then you can see we can get soybean oil very uniform soybean oil droplet through this microchannel. This is the oil droplet made by this microchannel emulsification. This is very unique technique to very uniform oil droplet. Using this uniform oil droplet, we try to modify the surface of this oil droplet by layer technique. This is a microscopic image of the oil droplet by microchannel emulsification. The average size is around 27 micrometer. The coefficient of variation, it's some index of the dispersity of the oil droplet is only 6.5. Normally, less than 10% of CV, coefficient variation, it's called monodispersed emulsion. But in this case, only 66.5 is very uniform, as you can see, oil droplet. And the zeta potential, it means the surface charge of the oil droplet is minus 65. It's very high negative charge of the oil droplet. Using this oil droplet, we modify the surface. We use the chitosan. Chitosan have the positively charged like this, NH3 plus here. And we use the carboxymethyl cellulose. We are calling CMC. It have this negative charge. So we use these polymers as coating materials of oil droplet. Firstly, we mixed with the chitosan. CHI means the chitosan. Then we get the chitosan layer on the oil droplet. Then washing out the remaining free polyelectrolyte, polychitosan, by removing the continuous phase solution and other buffer without chitosan. After that, we mix with carboxymethyl cellulose, CMC, then we get the CMC coated oil droplet. In this case, in this stage, there are two layers on the oil droplet. So next is just a repeat of the coating. Next is we add chitosan. Then we get the three layer chitosan, CMC, and chitosan. Then we add CMC. We get chitosan, CMC, 4 layers like this. Then we can repeat this step. Then we can control the number of layers on the surface of oil droplet. Then we can control the stability and release property by the number of the coating. This is a typical example of this layer by layer coating. This is the change of zeta potential. It means the surface charge of oil droplet by layer coating. First oil droplet highly negatively charged, but after the coating of chitosan, the surface charge changed to the positive because the chitosan have positive charge. after the coating by carboxymethyl cellulose change to the negative because CMC has negatively charged. So as you can see, the surface charge depends on the final coating material. Kaito-san CMC, Kaito-san CMC, Kaito-san CMC. So we can control the surface charge by changing the final coating material. Then this is a microscope image of oil droplet, and this is a fluorescence image of the oil droplet. We use the fluorescence label Kaito-san. So you can see the green fluorescence on the oil droplet like this. All of oil droplet covered by have green fluorescence. It means that oil droplet surface of oil droplet covered by fluorescence Kaito-san. So it's one evidence of the surface coating of oil droplet. We also observe the layer on the oil droplet by electron microscopy. This is the oil droplet microscopic image, TEM image of the electron microscopic image of oil droplet. You can see the size, relatively large. So when we enlarge this surface, we can observe this kind of layer. I think this is polyelectrolyte layer coated on this oil droplet. In this case, just 2 layers. It means the Kaito-san and CMC. But next slide, we can see the 6-layer coating. You can compare the thickness of the bilayer. Here is 2 layers, Kaito-san CMC 2 layers thickness. This is a Kaito-san CMC 6 layers thickness. So you can see the thickness of the polyelectrolyte layer seems to increase with the number of coatings. So it means it's possible to control the release of ingredient in oil droplet, and also it's control the stability of oil droplet. The one example of the stability enhancement by coating shown in this slide. This is the primary oil droplet. This is a Kaito-san CMC coating. This is a 6-layer coating of oil droplet. Then we wash out the emulsion in aqueous phase. Let's see after the removal of emulsion. In this primary oil droplet emulsion, most of oil droplet coalesce. They cannot keep oil droplet. They make just oil phase, not dispersed. But in the case of CMC Kaito-san coating and these CMC Kaito-san coating, even after the removal of emulsifier, we get oil droplet. It means this oil droplet can be stabilized these coating layer. So it's possible to make emulsion with emulsion-free emulsion using this technique. This is a short summary of the second topic. So the surface properties of large oil droplet were modified by electrostatic layer by layer mulch coating using cationic Kaito-san and anionic CMC. Then we can modify the surface charge and stability written like this. So this stability of large oil droplet could be designed by layer mulch coating using polyelectrolyte dietary fiber. I think these oil dispersion system would be useful for developing the new food and pharmaceutical product having improved control stability and target release. So another roughly 10 minutes, we talk about the last topic about the development of the in vitro human gastric digestion simulator for the direct observation and analysis of food or pharmaceutical drug digestion process. This is a little bit different topic compared to the previous two topics. So the background of this topic is the evaluation of release of pharmaceutical components and also the food digestion in the gastrointestinal tract is very important because understanding release of pharmacological compounds and food digestion provide us the basic knowledge about the controlling release or digestion and designing drug and food. For example, about the food digestion, this is a human gastrointestinal tract. So about the food, this is the life cycle of human. So for baby and elder people, more nutrition needed because the major or decrease of digestional level. On the other hand, for young people and middle-aged people, and also the some disease people, we can control the digestibility to control the nutrition intake. So this is the image of the human gastric intestinal tract and for our focus point. Normally, people take food, solid food. No, people can take the solid food. Then during the digestion, the breakdown and nutrition can be released. Then it can be taken up through the wall, human wall, gastrointestinal wall like this. So the observation of digestion process is very important to know how the food or pharmaceutics can be digested through the gastrointestinal tract. So then, but in the human, the experiment in the human body is restricts. So then we develop the in vitro human gastric digestion simulator we are calling GDS. So this is the illustration of stomach. You can see the movement of the wall of the stomach. This is the peristaltic movement. This is called the peristalsis. So this peristalsis affect the digestion of solid food in the stomach. So then our approach to develop the stomach simulator, considering the effect of peristalsis, because it must provide us the interesting knowledge about controlling solid food digestion. So this is a typical example of the in vitro gastric digestion system. People are using the frost shaking, or people are using this kind of vessel to examine the release of a drug or food digestion. But as shown in previous slide, the situation in the stomach is really different from this situation. So we made a human gastric digestion simulator based on the medical data of the stomach. There is a flow of the gastric contents here. So we use the simulator to mimic this flow in the stomach. Then we simply simplified the structure of the stomach, especially the antrum part of the stomach. Then we made this GDS model. So this is the photo of our GDS model. The peristaltic motion of gastric wall simulate using this roller to push the rubber wall like this. Then we simulate the movement of the gastric wall. Firstly, we try to digest the tofu as a model food. So we cut this tofu in 5 millimeter cube, then mixed with artificial saliva and artificial gastric juice. Then we put these in this gastric model. This is the This is the movie of the digestion of the tofu in the GDS model. This is immediately after the start of the digestion. You can see the tofu solid here. This is a cube shape. But after 100 minutes of digestion, you can see the change of the shape of tofu solid. Then after 3 hours, most of tofu is digested like this. So we think we can simulate the digestion of tofu in the stomach. And also, it's very difficult. It's not possible to see the digestion manner in human stomach, but using this DGS model, we can observe directly the manner of the digestion of food like this. So this is the, I will skip this one. This is the image of digested food in our model. You can see the tofu digested by using GDS model. The shape is very random like this. So it means the physical digestion occurred by peristaltic motion occurred during the stomach model. But when we use the Frasco shaking digestion as shown previous slide, just shake like this, the shape of the tofu is the same as the initial. I mean the shape is cubic shape even after the digestion. It means the Frasco shaking digestion, there is no physical digestion, so the tofu can keep the shape during the digestion. I think it's different from our real stomach. So then, this is a short summary of final topic. The human gastric digestion simulator GDS equipped with the peristalsis was developed. The shape of tofu particle after digestion experiment demonstrate that difference in the particle disintegration behavior between GDS model and Frasco shaking experiment. So we think, considering physical digestion induced by peristalsis together with chemical digestion, provide us new insight on solid food digestion and also the drug digestion in intestinal tract. So that's all of my lecture. So if you have some question about my talk, please send email or please send email to the professor. So I will reply for your question. I hope you interested in research topic and also interested in our master and doctoral program in our Tsukuba University. So thank you for your kind attention. That's all my topic.
Okay, thank you very much, Professor ICHIKAWA.
Yeah.
It was really very interesting topic for us, and actually there are many questions coming
Yes.
in the chat, but because of the time, so I think we will collect the questions and then we will send it by email to you. And
Yeah.
I hope you are willing to
I
answer the
will
question,
apply.
and then you just email it to me, and then I will distribute back to the lecturers and the students. Okay.
Yeah.
But the topics are really interesting.
Yeah.
You have talked about 3 different topics, but they are related to each other and also actually closely related with our research in
Yes.
Swiss German University.
Mm-hmm.
And some lecturers in our faculty, they are also doing research related with your topic. So
Yeah.
I think we can keep in contact.
Yeah. Of course.
In communications
Yes.
about
Then.
the research. Yeah.
Yeah, we can collaborate.
Yes.
Yes.
Okay.
Thank you very much.
Yeah. Thank you very much once again, Professor ICHIKAWA. And then for all participants, if you have questions, please write your questions in the chat room. And then Stasia will collect your questions and then we'll send it by email. And then we will send the answer to you also by email because of the limited time today. So actually the sessions usually delivered in 3 different sessions, meaning 3 multiply with 1 hour is in 3 hours. Okay. And we still have another session today. But before we start the next session, we will take picture together. So I hope you can open your video so that we will have a very nice picture together with Professor ICHIKAWA and Professor KANAMORI. And Stasia, can you help us to take picture? Okay, are you ready? So okay, Stasia, can you lead taking the picture?
Let's wait until all the participants open their video.
Okay, Professor KANAMORI, Professor ICHIKAWA, you can see in the faces of our students, alumni, and also our lecturers. Okay, Stasia, can you start?
I think, okay, there are 4 pages, so I will start from the first page. Please, everyone, have a big smile. 1, 2, 3. Okay, let's wait for a while. Okay, now we're moving to the second page. Everyone, 1, 2, 3. Okay, now to the third page. Everyone, 1, 2, 3. And to the last one. 1, 2, 3. Okay.
Okay, thank you. So we have 4 pages because today we have 78 participants. Yeah, I expected only 50, but fortunately we have more. And I would like to welcome also some lecturers I didn't introduce to Professor ICHIKAWA and Professor Kanamori. We have here Ibu Dr. Evita Legowo. Welcome, Dr. Evita.
Thank you, Pak.
Hello,
Yeah.
Nice to meet you.
Hello, nice to meet
Hello,
you.
nice to meet you. Good afternoon.
Yeah, nice to meet you.
And we have also Dr. Collis here with us.
Nice to meet
Welcome, Dr. Collis.
you.
He's
Dr.
also the director of research and community service in our university.
Nice to meet you, Professor ICHIKAWA.
Nice to meet you.
Nice to meet you.
Yes,
Good
and
afternoon.
we have also Dr. Maria as the head of food technology study program. Yeah, welcome.
Hello, nice to meet you.
Nice to meet you, Maria-san.
Yeah, she has graduated from Hokkaido University, Japan, in food technology. Yeah.
Okay.
And we have also Dr. Irfan. Are you here?
Hello.
Yes.
Yeah. Nice to meet you. Sorry, I
Good
cannot turn on
afternoon.
my video.
Good afternoon.
Good
Nice to
afternoon.
meet you.
Good afternoon.
Yes, Dr. Irfan has also a question to you, Professor ICHIKAWA. And
Okay.
he's a chemical engineer and he's now also our vice rector of Swiss German University. And we have also, I think, Dr. Dedi. Dr. Dedi, are you here? Dr. Dedi Wicaksono is head of biomedical engineering department in our faculty. And maybe there are also other lecturers here. I cannot introduce one by one, maybe. Okay, I think we can continue with the next session. So the speaker on the second session is Professor Toshiyuki Kanamori from the National Institute of Advanced Industrial Science and Technology, and also professor in the Tsukuba University. So Professor Kanamori, time is yours, please.
Can you see my slide?
Yes, we can
Okay.
see your slide.
So let me start. Good afternoon, everybody. It's my honor to meet you and give a talk today. I'm Toshiyuki Kanamori, a chief senior researcher of National Institute of Advanced Industrial Science and Technology, of which abbreviation is AIST. I will give a brief introduction about myself and AIST later. Okay. At first, I would like to apologize to you because some of the presentation materials used in this lecture are written in Japanese. But I hope you could understand them by figures. Here you can find the contents of today's lecture. First of all, I would like to show you my professional career briefly, by which you can understand the basis of my lecture. Then I will take a glance on our organization, AIST, and a technical view of our research subjects. I think you are familiar with the process of drug discovery and development, but let me make our understanding be on the same level, and I will point out the technical problems to be overcome for new drug launch. Finally, I will introduce our research topic as a new global trend in the drug discovery and development fields. At the end, I will wrap up my lecture. Because the time for this lecture is limited, I cannot give the detailed talk here. So please study furthermore by yourselves with reference materials shown on each slide. At the start of my lecture, let me show you my brief self-introduction. My major specialty is basically chemical engineering, which is same as Professor Tsukuba, Samir-san. You may be not familiar with chemical engineering, but this page shows the definition of chemical engineering declared by the American Institute of Chemical Engineers in 1983. When I graduated from a Department of Chemical Engineering, 1983. At that time, I learned very simply that chemical engineering is to design, operate, and maintain chemical equipment and plant. In the Department of Chemical Engineering, I was assigned to a strange laboratory for my graduation research study and master's thesis. The professor of the laboratory taught us that we can assume human body as a chemical plant, seen here in this slide. Taking heart as an example, we can easily imagine that the function of a heart is similar to a pump, like this. Let me introduce my professional career briefly. After finishing the master course, of which thesis was about hemorrhagic, I was working for a chemical company as an engineer and in charge of the development of new applications of polyester products. 5 years later, I left the company and re-enrolled the graduate school to receive PhD degree. The thesis for PhD was for the engineered design of hemodialyzer, that is, artificial kidney, and the optimal design of the therapeutic condition with artificial kidney. After belonging to the present organization, I have led many R&D projects from the point of view of the development of new materials and their utilizations to medical fields. Like this. This research subject is a bit different from the others, which has been done by Professor Tsukuba, Samir-san. Here, I briefly introduce my organization, AIST, and my concept to manage R&D projects. Do you know the phrase, valley of death in scientific research and development? This page clearly shows how difficult to utilize the scientific output in industry fields, telling the world valley of the death. AIST has the important mission to overcome valley of death, although it is too tough. We have so many actual R&D subjects which are related to the SDGs as a general rule. This data is for 2017 financial year, but you can know the size of AIST through these data. We have 10 sites overall Japan. The largest is in the Tsukuba city here. AIST is eagerly promoting global research collaborations with any type of research organizations university, company, and NPO like AIST. This figure shows international research collaborations with AIST under any contracts. We have made the research collaboration with Indonesia, as shown here. If you want further information about AIST, please visit this URL here. This figure shows my concept to propose and manage a research subject. To apply our research output to industries competing with another research organization, especially universities, I have established the research framework to handle everything by ourselves, from atom to device and system. Recently, we have several R&D projects focusing on applying microprocess with functional materials to drug discovery and diagnosis. Here I am moving to actual topics for today's lecture. Let's review the typical process of a drug discovery and development, which must be familiar to you. If not, find suitable textbooks, which is for undergraduate students, and learn by yourselves. Then I will talk about the recent global program to launch a new drug from the technical view. This figure shows the typical R&D process to launch a new drug. As you know, before and after clinical trial, the process is divided as the drug discovery and drug development. Recently, drug discovery is called as non-clinical trial. If you don't know the detail for each step in this process, learn it by yourselves with textbooks. In addition to conventional technologies using enzyme, cell, and animals, recently many computer-aided technologies such as bioinformatics, in silico biochemistry, and system biology have been utilized. You know, many papers published in the international journals have pointed out the crisis for new drug launching. This figure is a typical result. If you are interested in this topic, why don't you read this paper? Anyway, it becomes extremely difficult to launch a new drug nowadays. They say drug discovery and development are kind of gamble. In addition to that, it becomes much more difficult to use animals for experimenters. In European Union, after 2013, any cosmetic products developed using animals have been not able to be sold. Therefore, to find any alternative technology to animal experiments is the most important and urgent issue for cosmetic and chemical companies. This figure shows the famous cliché, a dilemma for drug assay, which is the most serious problem to be overcome in drug discovery and development. In recent years, many kinds of human cells are utilized in drug discovery, but not sufficient. I mean, the result obtained with the human cells culture system cannot be always related to the results obtained in clinical trial. Reducing experimental animals is a global tide, but they are still now needed. But they are still now used in the drug discovery, especially to be approved for clinical trial. However, the results obtained with animals are insufficient regarding to the validity, and animal experiments are troublesome and sometimes extremely high cost. To overcome this dilemma, we need a novel technologies which can mimic the drug functions in human body. In the last part of my lecture, I will introduce one of my R&D subject, microphysiological system, MPS, which is one of the hottest topics in the drug discovery and in the world. Do you know this person? Anyone know this person? He is Alexander Fleming, whom everybody listening to my lecture must know. He is a Nobel Prize laureate as a discoverer of a penicillin, but he had the other many achievements in biomedical field. For example, the discovery of lysozyme here. This photo was maybe taken about 100 years ago, but you can find the typical biotechnological instrument here. You know, there are petri dish. I have looked up that the Petri comes from the name of the biologist, Dr. Petri, who was a student of Professor Koch. Koch is, as you know, the founding father of modern microbiology. I think petri dish was developed around 1890. Therefore, we are using this instrument over 100 years. Although recently, we using a multi-well plate like this, but basically the same. It's an amazing fact. Can you name another technology used for over 100 years? I think nothing. This figure shows how we can look at the biological cell from the point of view of chemical engineering, which is my major specialty, as I have let you know. If we can regard a biological cell as a chemical reactor or chemical plant, we can know we have to re-examine the environment of a cell culture using existing instruments such as petri dish and cell culture flask. This photo is of typical cell culture. As you know, there is many and huge discrepancies in the environmental around the biological cells between in vitro and in vivo. For example, epithelial cells are generally used in biomedical field, but epithelial cells have many morphology. In vivo, as shown in this slide, and as you can see this slide, when I was a student, we thought the function of a liver was based on hepatocytes, I mean, we didn't know the functions of non-parenchymal cells, parenchymal cells such as uh, uh, stellate cell, stellate cells, and dendritic cells. However, recently, as the functions of non-parenchymal cells become clear, we know non-parenchymal cells are necessary to keep the function of liver. The discrepancies are listed here for examples. Anyway, because the magnitude of a biological cell is several tens micrometer, microprocess is promising to precisely control cell culture environment. Okay. Here, I change the topic a bit. Do you know the technical term named MEMS, which is an abbreviation of mechanical systems? Do you know this technical term? In this plot, taken with scanning electron microscopy, same, you can find small insects, thick. And here you can see very small gears. They can move actually. Amazing. This work was just for demonstration of the usage of MEMS. Early 1990s, the technologies which have been developed in the semiconductor products were called as MEMS. Okay, this MEMS technology have been developed in the semiconductor products and tried to be applied to the other fields. At first, chemistry, especially analytical chemistry. This stream is related to the keywords lab on a chip, laboratory on a chip, lab on a chip, and microtas, micro total analysis system. In this decade, MEMS is being applied to biology, especially to handle biological cells. While at the same time, we had many studies concerning to human stem cells, leading to deeply understanding human cells and the various technologies in practical use to handle human cells. The combination of a MEMS and human cell application bring a new technical concept named as microphysiological system, MPS, as shown in this figure. The basic concept of MPS is to mimic the in vivo function of an organ or tissue by utilizing a microprocess to precisely control the culture environment of human cells. So this concept was called as organ on a chip in past days. MPS is one of the hottest topics in drug discovery in the world. Therefore, many excellent research groups in the world are competing concerned to this subject. If we can derive the in vivo functions of several organs, we can mimic the interaction between the organs. This concept is called as organs on a chip. Organs on a chip is expected as a promising alternative technology to experimental animals and can promote drug discovery and development by enhancing the predictability for drug development in drug discovery, leading to the dramatically reduce of the drug development attribution. To realize this technology, the combination of a microprocess, process engineering and cell biology is very crucial. Because organs on the chip can mimic the kinetics of a drug in human body, organs on the chip is a promising alternative technology for experimental animals. This movie is summarizing. This movie is summarizing my talk about MPS. Okay. あれ、動かないか。Wait a moment, please. Okay. This is the in vivo phenomena. This is organs on the chip as a wet in vivo simulator. Okay. So let me wrap up my today's lecture. I would like to give you a take-home message, which is not directly related to today's my topic. As you know, they say the 19th century is called as chemistry era. Chemistry drove alchemists away. Chemistry drove alchemists away. I mean, chemistry era was also dawn of natural science. And 20th century is physics era. I mean, in the present days, chemistry can be explained with physics. Now we have the 21st century, which is called as bioscience era. This photo is of an artistic work named elixir, which means a drug to give an eternal life, non-limit life. Even at present, I mean, our society must be science-based. sometimes I have the opinion that a living body is special. However, as far as the living body consists of molecules, we must explain all phenomena in the body by chemistry and physics. Although you may not know this Chinese character, even Samuel-kun, Professor Kusumo can not understand this Chinese character. We call Chinese character kanji in Japanese. We can pronounce this kanji character as kotowari. I'm very sorry, but I cannot find any suitable English word related to kotowari, this character. I think reason, rational approach, logical manner, and so on may be near, I think. Anyway, in your future study, please keep this taking home message in your mind. We have enough time. So if you have any questions for you to ask me right now, please click the hand raise button. If not, I'm waiting for your questions and comments by email at this email address. Thank you for kind attention. Terima kasih. Thank you, Samuel-kun.
Thank you very much, Professor Kanamori, for the very interesting topics. So it is very comprehensive topics related with chemical engineering, biochemistry, biochemical engineering, biomedical engineering, and everything's related with physics, chemistry, biology. So I think it's a good knowledge for all of us as students and also lecturers. And actually, we have a question here from Dr. Collis, Professor Kanamori. I don't know whether you want to answer directly here in the Zoom or you want to answer by email. So basically, Dr. Collis is also interested in the drug discovery. He's doing also, maybe Dr. Collis, we still have little bit time. Can you explain what are you doing here in the Faculty of Life Science?
Thank you, Pasam. Thank you very much for the opportunity, Professor. Indeed, very interesting topic especially for myself, because I'm also working on drug discovery, especially from natural products. I'm also planning to work on nano encapsulation of these natural products, and we'll have also a webinar next week on Thursday. So I was just wondering whether you are also working on, well, you mentioned about kaitosan, yeah, one of the very famous popular topics nowadays in nanomaterials. So I was hoping that we can collaborate in the future for our research. And you know that natural product, many natural products are non-polar substrate or non-polar compounds. So how we can make it as even more susceptible or for drug discovery purpose. So I think it will be interesting for myself, and also maybe you will be also interested in the research. And also another one, I'm also looking for high throughput screening technology, because I'm working on natural product library. So if we have a large number compounds or extract or fractions collected from natural products that can be screened by using this technology. So I'm again very interested to work together with you in the future, and also you can give me some information about that I'm working on. Thank you, Prof. Thank you, Pasem.
Thank you, Professor Kanamori. So basically, we are interested in doing research together with you because your topic is very interesting.
Thank you.
Yeah.
And but I have not focused on the high throughput screening, but high content screening. Okay, high content analysis, because as Professor Corris knows, ITS made high cost for drug discovery recently. So they need to HCA, high content analysis or high content screening rather than HTS to reduce the cost to develop new drug. So we are focusing on the HCA or HCS rather than HTS.
That's also very interesting. So I didn't know about that before. So my knowledge is quite limited for new technologies. So, yeah.
Of course, at the present, HTS is a very important in the discovery and drug development, especially drug discovery. But
thank you very much. Yeah.
But they want to reduce the cost to launch new drug. So HTS makes high cost to find collaborate a promising candidate. Even use HTS. Okay. So researchers working in the pharmaceutical companies do not want to more high throughput. More high throughput. No. So they want high content. But of course, balance is important. Content and throughput. The balance between content and throughput is important. But okay.
Thank you, Prof.
Yeah. Thank you. We have also here Dr. Dedi, Professor Kanamori. Dr. Dedi has taken his master degree, I think, in Japan, and then doctoral degree in Netherlands. He is also very interested in these topics. So maybe, I'm sorry. I have to turn off my video because there's something wrong with my connection. So Dr. Dedi, can you tell us a little bit about your research? Maybe you are doing also research on organs on chip.
Yes. Hai Kanamori sensei, Dedi tomoshimasu. Toko daishu desu.
Konnichiwa.
Konnichiwa. Watashi wa moto Aizawa sensei no.
Hai, よく知ってます。
Yeah. Yeah, I will continue in English so that everyone can understand.
Okay.
So yes, we have also some research in microfluidics here at STU, Swiss German University, even though very simple one for diagnostic purposes. And yes, I have heard about this organ in a chip and organs in a chip. If I may know what types of cell culture that you are doing, that you have been doing there at AIST, and maybe you could advise us on simple cell culture that maybe we can also start here at our university, because we don't have yet very advanced facility to do cell culturing. but perhaps we can start somewhere. Kang Tangna system mesti areba, so that we can also start somewhere to also do this or to start organ on a chip research.
So, as you know, I have prepared my presentation materials today to give a talk to the undergraduate student. So I have to skip my actual research output today. But if I if I want to talk about a detail about MPS system, I need maybe 3 hours, 5 hours. So as you know, I and Ichikawa sensei and Professor Kahayo were planned to visit your country and your university in maybe today or in this week. So if we have time to meet you, actually, I can discuss with you. And it's okay to set another web meeting with you to discuss more deeply about today's MPS situation in the world, telling more and more detailed research outputs, including ourselves. And I checked many other research sites, and I have many information about their research output. So if you could have a time to discuss with me, I can tell you more detail the situation about the more detail about MPS situation in the world. And actually, I am now the principal head of the national project to develop MPS system, very huge national project. So we had made many research achievements to concerning about MPS system. Maybe my research group is one of the hottest site to develop MPS system in the world. So why don't you have another chance to discuss with you about this subject? It's okay?
はい、そうします。はい。イメール連絡したいと思います。
はい、thank you.
You're welcome. Thank you very much.
Pak Dedi can speak Japanese very well. Okay.
Little bit, Pak.
Thank you, Pak Dedi. So actually here we have also Pak Victor from company. Pak Victor, are you still here? Maybe Pak Victor from PT Deltomet. Oh, maybe, okay, he cannot hear my voice. Okay, no problem. So we have also Pak Victor is Director of Research and Development in Deltomet Laboratories. They are developing herbals. And then we have also here Pak Heri Sutanto. Actually, he's chemist, but he became chemical engineer now because his doctor degree is in chemical engineering. So he's interested in the droplet presented by Professor ICHIKAWA. And I think we can work together. And then we have also Dr. Irfan Kartawirya. He is also chemical engineer. He is also interested in the presentation of Professor ICHIKAWA. So Professor ICHIKAWA, do you want to answer by email or do you want to say something maybe?
Maybe there are a lot of questions. So
a lot of questions.
I will reply by email.
Okay, yeah, because this is very technical. Yeah, no problem. And we have also some question from the students. I think we will send an email to you.
Yeah.
And also from Miss Stella. Miss Stella is from food technology. She's now taking a doctoral course in Osaka University. She thanked for your nice talk and was interested also in the GDS model. Yeah. And then we have also Miss Elga Saki. She is a student of biomedical engineering. Will be graduate next year. She's now preparing the research proposal. She's also interested in your talk about the lipids, yeah, coated with multi-coating layers. Yeah. Okay, there are many questions basically, but because of the time, so we usually we finish at 4:50, and now it's already 4:48. So I think I have to close this guest lecture now, and I would like to thank Professor Kanamori and Professor Ichikawa for giving a very interesting lecture to students, lecturers, and guests today. And actually, Professor Kanamori and Professor Ichikawa had a plan to visit Indonesia this year, as I mentioned, but because of the pandemic, and they had to cancel the plan. So I hope next year the condition will be better, and then I hope next year you can visit Indonesia.
Yeah.
We hope it will be better. we believe.
Yes, and then you can visit SU. Okay, thank you.
Thank you.
Once again, Professor Kanamori and Professor Ichikawa, and thank you also for all participants, students, alumni, lecturers, guests, for attending this special guest lecture today. I believe you have learned many things, new and important things related with drug discovery and so on. And once again, if you have questions, please write your questions via the chat, or you can email to me or to Stasia, and then we will forward your questions to Professor Kanamori and Professor Ichikawa, who will be gladly answer your question. Okay. Once again, thank you, and see you again next time. Have a nice weekend. Stay safe and healthy. Arigato gozaimashita.
Terima kasih.
Terima kasih.